As we conclude day 3 here at the ASGCT annual meeting, I'm joined by Dr. Todd Carter, Chief Scientific Officer at Voyager Therapeutics. Voyager has had quite a journey since its founding about a decade ago, and Dr. Carter has been there for a good part of that. At this week's annual meeting, Voyager had 12 presentations focused on updates across their AAV capsid discovery platform supporting various CNS gene therapy programs. Now, Dr. Carter, Voyager revealed earlier this year a new partnership with Novartis, which includes work on Huntington's disease. And just this morning, Bev Davidson shared an update on her current work unlocking AAVs for CNS disorders, also including Huntington's. And I know there are other programs in development across academia and industry. That's great news for patients, especially after past false starts. Broadly, what's changed with the underlying technology or approach that's enabling this resurgence in gene therapy development for diseases like Huntington's that are rooted in neural circuit disruption?
First, I'd just like to say thank you for the opportunity to talk. I'm really excited to be here at ASGCT this year and to see all the amazing progress that we're making across the field. Regards to your question with Huntington's disease in particular, and really the field in general, I think it's about delivery. Really the ability to deliver broadly to the particular regions targeted in a disease. And you mentioned the particular circuitry involved in Huntington's disease. So Dr. Davidson's talk this morning really went into some details around a novel capsid that she has identified that when delivered into the CSF is able to distribute broadly throughout the brain and to the right regions. And that's what's enabling her and for us in our collaboration with Novartis are novel capsids that allow us to get into the brain and the rest of the CNS after an IV delivery, penetrating the blood-brain barrier, are really opening these new doors. That's the first part. The other part is in the expansion of the therapeutic targets. And so one of the things that we're approaching is not just targeting Huntington's, the gene itself, which we are, but also expanding into some of the other genetic modifier diseases genes, such as MSH3, which may be involved in the genomic expansion. So really it's twofold. It's the targets involved in the disease and the ability to deliver to the regions that are impacted by the disease.
Across our field, we're getting closer to realizing the potential of CGT applications for larger disease populations like Alzheimer's and ALS, which I know you all are also working on. And with new research like that coming out of the Sant Pau Memory Unit in Spain this week, positing that 15 to 20% of Alzheimer's cases could be directly caused by genetics, what are implications for CNS gene therapy developers like Voyager?
So I'm a geneticist by background and training, so I'm always really excited by new genetics. I think what we're seeing here is an expansion of what was already known about ApoE and the genetic links to disease. And really we're starting to see it more and more as a causal disease gene, the ApoE4 allele itself. And the dose response that we're seeing with the number of ApoE4 alleles and the impact on the likelihood of disease is really making it look like a disease-causing gene. The importance of that is a couple of fold. One, it helps us define a population that will be affected and unfortunately for the patients, affected more quickly. But interestingly enough, the course of the disease looks to pretty much mimic or be a standard course of what we might think of classically as a sporadic Alzheimer's. And so it's a patient population we can identify and then do our clinical trials and to test our therapies. The other component of it is that now that we can identify them, that's a group that we can think about starting earlier and testing these treatments in a population for which we really suspect they're going to have a benefit. So it's in their best interest, in our best interest, to work together on those to try to get those clinical trials done.
And as we've been discussing, there are many emerging modalities for CNS gene therapies. Why does Voyager believe that the future of CNS gene therapies lies in AAV capsid engineering as opposed to other types of vectors or focusing on the payload?
Well, I think it's both those things, or all of those things. So it's the ability to deliver, which I spoke quite a bit on before, and it is on the payload as well. We're going to need more and more sophisticated payloads to target these different kinds of diseases. And when we look at the larger disease indications, those of the larger populations, it's going to become more and more important that we're able to basically thread the needle of the risk-benefit ratio. We're looking at multiple programs in Alzheimer's disease. We have a tau knockdown program. A lot of that work is being led by an ASO approach right now that's in the clinic and looking very good and very promising for knockdown of tau for Alzheimer's disease. What we're trying to do is take our novel capsids that allow us to deliver after a single intravenous dose and effectively use that same mechanism to knock down tau and have an effect in the disease. So it's a combination of the kinds of payloads that allow that and that delivery. And I know I'm being a bit repetitive in saying it's both those things, but I do feel pretty strongly. And also, just additionally, it's not only AAV gene therapy. I think there's a ton of work that we're seeing all around this conference. There are different ways one can deliver, and the ability to deliver, regardless of whether it's AAV, which it's near and dear to our hearts, but there are other methods to do so as well that I think will be really important for these diseases.
And on that note, we will conclude our Day 3 Sound Bites. Thank you, Dr. Carter. And if you missed any of the sessions, including Voyager's presentations, you can catch them on the app for 30 days on replay. All right, see you tomorrow.